Hello everyone. Good morning, good afternoon. Welcome to today's webinar, Ongoing Trials During COVID-19, Quantitative Strategies and Methodologies. It's my pleasure to moderate this webinar with a great expert panel of regulatory and industry experts. They will be discussing challenges of trials impacted by evolving COVID-19 pandemic, and they will review decisions to be made regarding the collection, review, and interpretation of the data. My name is Natasha Müllerman. I'm Vice President of Strategic Consulting at Cytel. And it's my pleasure to moderate this webinar with a great expert panel. And now they will present themselves.
Hi, my name is Greg Campbell, and for 20 years I was the Director of Biostatistics in FDA's Center for Devices and Radiological Health, CDRH. I have a PhD in statistics, and before that, and spent some time at NIH before my 20 years at FDA. I left FDA about 5 years ago and now function as an independent statistical consultant.
Hi, I'm Simon Day. I'm a statistician. I've worked a bit in academia, then moved into industry for a number of years. I then moved to the Medicines Control Agency in London, as it was at the time. I was head of the statistics unit there and also managed one of the clinical therapeutic groups. Did a lot of work at the European Medicines Agency, particularly vice chairman of the Scientific Advice Working Party there. And I now work as an independent statistical consultant as well.
Hi there, my name's Paul Terrell. I also work for Cytel like Natasha within the strategic consulting team. I've been a practicing statistician for 20 plus years now. I actually started in the agrochemical world, working with formulation chemists before moving into pharmaceuticals. I'm what I'd call very much a traditional statistician. I still like to get my hands dirty, very much into the actual experimental design and subsequent inference of studies.
Hi everybody, I'm Rajat Mukherjee. I'm a statistician at Strategic Consulting Cytel. I have a PhD in statistics. I started my career in public health, designing observational studies, and then around 10 years back, I moved into industry, looking at clinical trials and adaptive designs and Bayesian designs, and also looking at big data problems.
Hello everyone, my name is James Matcham. I'm a senior principal in the strategic consulting group at Cytel. I have over 30 years experience in the industry. After a short academic stint, I worked at Amgen for over 20 years as a statistician in the development of their drugs. And more recently been the Vice President of Early Biometrics and Stats Innovation at AstraZeneca.
Thank you for the introduction. I'd like to start the discussion with the first question. The limited access to hospitals and also the healthcare professional focusing on critical tasks, plus the fact that many patients would have to self-isolate. All this results in changes in study visit schedule, also missed visit, or even patient discontinuation, which in turn leads to missing information. Could you share your ideas about strategies to proactively minimize missing information or manage this missing information?
Okay, thanks, Natasha. Perhaps I'll take that first. I've been astounded by the scale of the number of trials that have been paused. I read recently that nearly 500 trials in the US alone have been paused over the last weeks because of COVID-19. And I'm quite sure this pattern has been repeated around the world to a different extent and perhaps different timings in different countries as well. With patients not being able to attend centres because they're isolating, or centres being closed as they focus on COVID-19, it's very clear the impact of COVID-19 on ongoing trials that are there right now. I think it's clear that as we come out of the COVID-19 pandemic, all trial reports will contain a section in the report that will talk about the extent of the impact of the COVID-19 pandemic on the study. And this will include contingency methods, data collection methods, the influence on the results and the conclusions and so on. And as we run these trials, we should be getting ready to be writing those sections. It will be very clear and important to collect data on the local timings and extents in the different regions of the COVID-19 mitigations that are taking place. But also in the studies, we'll have to collect individual reasons for individual patients about why data is missing, what has happened, the protocol deviations, and collecting data at that individual patient level is the task that's in front of us all. So as we run a study, we'll have to identify patients as either being affected or unaffected by the COVID-19 pandemic. And I wanted to talk about those patients that have been affected. I think that in ongoing studies, they can be affected in quite a few different ways. But the two that I'm going to pick out are those patients that have not been observed for some reason, and those patients that have not been treated. And there may be patients that have not been observed or treated. So for patients not being observed, it's because they've had to pause the traditional visits to the centres. And one of the questions that was put forward was, so what do we do? If patients can't do the traditional visits in the centres, how do we mitigate that? And so there's been some discussion about visit conversion. So how do you convert a traditional visit to something else? And to ensure the safety of products, I mean, that could just be a simple phone call. It could be a virtual visit, a video visit, or it could mean the patient going somewhere else to get their observation, to get their samples done. So it could be a local lab, it could be a new vendor, it could be staff visiting the patient at home. And what's clear is that when we report a study at the end, there'll be patients that have had visits converted to these new styles and those that have had traditional visits. And I think that that's one aspect that we'll have to talk about in the reports, is what's the difference between data collected during a traditional visit and the data collected during a virtual visit. And I think there need to be some aspect in the report that talks about how they compare with each other. So one aspect might be that imagine that you decide to collect data from a patient using some sort of virtual methodology, is that as you bring that into a study to try and mitigate for not having traditional visits, that as we come out of COVID-19, that you might continue to do that through the study, so that at the end you could do the comparison of traditional and virtual methods of collecting as you come out. So trying to mitigate the fact that patients can't be observed.
James, can I ask you a question on that?
Sure.
I agree completely with you about getting patient-level reasons. It's not just a bland COVID-19 happened and so things were missed. It needs to be patient by patient. One of the things that worries me, and I'm afraid I don't have a solution, but I'm concerned that if we didn't have COVID-19 going on and a patient was due to go for a clinic visit but didn't go because they felt so awful, so dreadful, they really just couldn't face leaving the house. So we have missing values there. And we might well think about them rather cautiously and perhaps maybe do some imputation or something, and it would be on a conservative level, thinking that maybe perhaps this patient's not so well. But now we've got this identical patient who's due to have a visit today, clearly is not going to go to the clinic because of the current situation, is feeling absolutely dreadful and would not have gone to the clinic anyway. But actually we now get some information from them. And of course, we might capture the fact that they're really not feeling very well. But I actually have a concern that some of the missing data that we now get, we wouldn't have got, and we're not going to be able to tease apart. If in some ways, we'd like to know which visits would have been missing had we not diverted to a telephone system or some other system collecting data.
That's a really interesting point. I mean, you're assuming that the patient who's not coming in because they're feeling awful perhaps might have coronavirus. I do wonder that most of the patients that are not able to coming in are probably fine, or if they do, they're asymptomatic. So I think there's distinguishing between patients that are actually suffering from coronavirus and the vast majority of the patients who just won't be able to turn up to the site because it's closed.
Yes, I think I'm more concerned about the patients who are feeling very ill with the underlying condition you're trying to treat, and so wouldn't have turned up to a clinic visit. But it's just another one of the difficulties to try and handle.
Yep, definitely. So other mechanisms for pausing traditional visits, apart from converting the visit to something that's virtual, might be to be a little more generous about when a visit counts. So imagine that a patient is on a 6-month study and the two months in the middle is missing because of the COVID-19 pandemic. It may well be that in the analysis plan, windows that we had for certain visits can maybe relax quite a bit more, just to be able to, include patient visits as they might be. But again, that would mean a change in the protocol and perhaps analysis plan to be able to do that. So extending windows is another idea to do that. The other aspect about pausing traditional visits is that if you have a composite endpoint, so there are 5 or 6 parts of the composite, that there may be some parts of the composite that are very readily available even though the patient's not able to come into the site, but there may be parts of the composite that are not available because the patient's not been able to come into the site. So you may only have partial information on the composite. And again, how would you deal with incomplete data on a composite endpoint? Again, that is something which I know we'll discuss later as we talk about analysis plans and changes to protocols. So the patients pausing the traditional visits, we can try to convert visits to virtual ones, particularly to assure the safety of the trial participants as we review their safety. But I think it does bring lots of other issues about missing data. We'll certainly have to recall the reason for the missing data and then dealing with that in the analysis. So one aspect then of COVID-19 is not being able to observe the patient. There is another one, and that's when a patient is not treated. So for some reason, the treatment is paused or the treatment is actually withdrawn. And I think that that's another aspect. And are there things that we can do in studies, ongoing studies, to allow for pauses in treatments? If you imagine a patient on an active arm maybe pause the active treatment, but then may have to get the standard of care for a few months while that patient is being treated still as part of the trial, and then may then go back onto the active treatment as the pandemic lightens up. So you could have treatment switching going on through the study to be able to keep patients on trials. And again, that is another aspect of trying to keep a patient practically on a trial. So most protocols probably won't allow holidays from drug. So there may need to be protocol amendments if that's how it's going to happen. There is another aspect of keeping a patient on treatment if we can. And that is if the treatment is self-administered in any way, can the patient self-administer at home and the drug be delivered at home? And so again, that may be another practical way of keeping patients on treatment.
We have some questions.
Going back to your proxy home visits, in some instances, presumably we could use a proxy endpoint, particularly if we can use some form of patient-reported outcome as a proxy to what was going to be measured in the clinic. Not always possible, but there are situations when we can do that.
Yeah, and there may well be digital versions of traditional endpoints. So I'm aware of things like 6-minute walk test. In a normal clinic visit, that patient would physically do the 6-minute walk test. It may be possible to give the patient some digital device which then approximates their 6-minute walk test, over a period of time. So that may be possible to substitute that, but that would have to be done quite quickly to be able to mitigate the missing data that may occur otherwise.
We have some questions which came from the audience. So I will read the question out for the panel. Question 1. For those studies which are impacted by COVID-19, should the statistical analysis plan containing a section about the COVID-19, such as impact on data collection, handling the data, implications, etc.?
Yes.
Yeah, I was going to say the answer is yes. I mean, how you do it is dependent on how you write analysis plans. You could have one section that covers various bits, or you could insert it into the relevant parts of the SAP. But for sure, in a lot of these studies, we are going to have to update these things and reference it.
Very simple
Actually,
example.
I was
Sorry.
So I was talking to a company this morning about this very issue, and we decided that the best thing to do would be to keep the SAP as it was. It was all signed off, but to add an appendix to it. So it would be very clear, what the sort of the pure SAP is if this issue hadn't occurred, but still as part of the SAP document, but as an appendix. These were all of the special issues that were taken into account. We thought otherwise it would get really quite confusing to look at the SAP, because you could look back at past versions of it. It would look confusing to look at the SAP and try and figure out what are the special things introduced because of COVID-19, which might be missing data approaches or something like that. So our view was have the SAP as was and an appendix to go with it.
Just adding to that, a very simple example is, I think most of you and in the audience have thought about it, is a temporally stratified adverse event analysis. So look at adverse events pre-COVID, during COVID, and post-COVID. Hopefully we'll see post-COVID soon.
So my take on the question is that absolutely there needs to be an update to the SAP, and having it as an appendix seems like a really nice idea, Simon. I think it's clear that when we write reports or studies from now on, that there will be a section, and that section will be talking about the contingency measures that were put in place to deal with COVID-19 issues, lists of patients and data that were influenced, the affected data, and specific analyses that were introduced to either look at comparability of different ways of collecting data, or new analyses to try and understand the influence of the missing data on the conclusions. So almost definitely there will be updates to the SAPs. But just to flag that I think there are lots of other trial plans that should be updated as well. To name a few, the study monitoring plan will clearly need to be updated. The data management plan, any data processes, any data flowchart plans that you may have in a company, and the protocol and the analysis plan as well. So I think there will be many plans that will be updated.
Thank you. We have another question. How do we account for potential drug-drug interactions on those used to mitigate symptoms of the COVID-19?
No one wants to take that.
We're going to report adverse events, and there will be, certainly for serious adverse events, there will be some comment about whether it was thought to be associated with something else that the patient has given, which may be for COVID-19 or it may be for anything else. So I think that's going to be something to discover along the way.
I think we'll also need to look at some sensitivity analyses because I know it's not commonly done having time-varying covariates, but I think there would be some sensitivity analysis needed with using time-varying covariates.
I mean, obviously, it's critical to capture the information. Everything that James said, we've got to make sure we're capturing all the appropriate information. And it may depend how often it happens and whether it's the same common drug that's being introduced in parallel. The more data we have on the same sort of drugs, the easier it is to try and pull apart the information. So I think it depends on what else is being given and how common it is in terms of being given.
And another aspect is that if any drug is being given for COVID-19, presumably it's experimental. And I think that if you've got an experimental drug, most study protocols have as an exclusion criteria the use of another experimental drug. So there's a good question about, if you've got a patient on your study that you don't want to stop and you don't want the patient to leave, should you allow that patient to have an experimental drug for this for their coronavirus? That's actually a very interesting question, and I'm sure study teams will be thinking about that.
We have a lot of questions from the audience, so I think we'll try to also address them as we move forward. Maybe I will put the last question to the panel in this section, and it probably could merge us and bring us to the next question of evaluating impact on efficacy and safety assessment regarding protocol modification. The question is, how would you recommend to deal with missing final visits, which occur during the period to pause when the primary outcome needs to be done in person?
Yeah, I mean, just a small comment that intermediate visits in some sense are less important, at least for efficacy. I mean, they're often there for the purposes of keeping patients engaged in the trial and certainly for monitoring for adverse events. But in terms of the efficacy measure, losing one midway through doesn't matter quite so much as losing the end one. I think it's going to be a missing data problem. And it gets back a little bit to my comment that I asked James about earlier. If we know that the only reason the patient has this missing observation is because of COVID-19 and we're not involved in a respiratory trial or something like that, then we may well say, well, we're reasonably happy with missing at random or missing for sort of administrative purposes. But if there was a potential that those data, even if COVID-19 were not happening, would have been missing anyway, because, as I suggested, perhaps the patient was feeling so awful they didn't want to go to the clinic, then that needs to try and be captured, I think.
But it's not, I mean, we often focus on the final visit for our assessment, but there's a lot to be said for making use of the longitudinal data that you collect during the study. In fact, if you only ever focus on the final visit, you can throw information away, and this is whether you're within the COVID situation or not. So we have a lot of other information that can be used either as part of a MMRM type model or helping to build into multiple imputation model. It depends on how the objective has been phrased and what the actual efficacy assessment is. If you're treating someone through a year, it's not just where they are at week 52, it's where they are during that year as well.
It depends what your estimate is, doesn't it?
Exactly.
It depends on them.
Yeah, so it's actually a very nice link to estimate, because we have a question to the panel. Could you comment on the COVID-19 impact on estimate?
Yeah, so I'm going to take this one because I quite like estimands. I think it's fair to say that the consequences of COVID weren't planned, and we didn't really consider this sort of major thing was going to happen. However, they are going to have a big impact, and they're going to potentially impact several areas. So, and James has touched on some of this already, but they could impact on what we're measuring and how we're measuring it, whether we have to change how we're measuring it. So they could impact the variable of interest. They could impact the population in our study. You could imagine certain parts of the population are more affected by this, maybe the elderly, maybe the young, maybe the more severely ill could be affected more. So the population behind the trial can change. And we've already touched this as well, James has mentioned it, the actual treatment we're giving the patients could change. We've talked about treatment breaks and treatment switching. We've talked about additional medication being given. And those three attributes I've just described, the variable being measured, the population for treatment, are exactly what come out of very nice new ICH E9 addendum that focuses on these estimates. So we can take the addendum of the estimates and pull apart the different attributes and then try to work with them and work out what exactly it is we're trying to estimate in this study. Now, as I said in my introduction, I'm very much a traditionalist, and whenever I work on any experiment, whether it's a clinical trial or outside, the first thing I do is focus on the objectives. What is it we're trying to achieve from this clinical study? And by focusing on the objectives and whether we need to modify them, we then start to drill down to the effect and the impact of what we're doing actually has on the efficacy and safety assessments. But we need to know what always go back to what question we're trying to answer before we can then assess the impact and things we're going to have to change. So we may need to read it. Yes, go, Roger.
Just a question to you. You start with looking at the objectives, right? What are you estimating? How are you estimating? What is the population? But then you didn't plan for COVID, and COVID comes along. And the definitions of those things, or the interpretations or the assumptions, they sort of change, right? And the question is, how do you look at the impact? How do you measure, what is the impact? And that has consequences on, all sorts of decisions.
Yeah, that's right. So we have to, well, first of all, we decide whether we need to modify our objectives, and we're allowed to or not. But if we want to stick with our original objectives, we need to, there are different frameworks. For example, I'm sure that you've heard of the hypothetical estimand, where you try to estimate what would have happened if COVID hadn't come along. And you can either do it, a variety of ways of doing it. One way is actually to artificially create missing values. So some of the data we've collected, we choose not to include in our analysis because it could have been affected. It's linked to what James says. We need to identify data that may have been affected by COVID and data that may not have been unaffected. And if we can do that, the data that is effective, you could treat it as missing for your primary analysis and use imputation methods based on borrowing from unaffected data, both of the patient itself and from the other patients. So we could go down the route of what would have happened if this hadn't happened, if you see what I mean. Or we have to still say, well, I'm going for more of a treatment policy type estimate. Actually, this just has happened. I've still got an active comparator or placebo comparator. Okay, now we're in a slightly different situation, but I can still compare my two treatment arms. Under the assumption, it's a major assumption, that the COVID-19 hasn't impacted one arm differently to the other, you perhaps still got a valid treatment comparison. It gets difficult if there's some form of interaction between a treatment effect and COVID. Then it's harder, to now take that approach.
Paul, as you have mentioned that you like discussing estimate, I think the question from audience probably is for you because it's an estimate. Is the intermittent missing visit a missing data issue, or should it be addressed as an intercurrent event in the estimate strategy?
Well, sometimes intercurrent events create missing values, so you can't always pull the two apart. Or missing values are due to intercurrent events. So I don't necessarily separate them out. And it comes to what you want to estimate, whether you want to use that data or not. As I say, sometimes, is it missing? You've collected something, but have you collected what you wanted to collect? And if you decide you haven't collected what you want to collect, you can treat it as missing. If you think what you've collected is still of use and contributes to the analysis, then I may still make use of it, even though it follows from an intercurrent event. I don't know if that helps answer the question. If anyone else got anything to add to that?
We have another question about the analysis. How to define the analysis data set such as per protocol and modified, looking at the current scenario?
So this is an interesting one. First of all, I must admit, I'm not a fan of per protocol analysis sets anymore. Since the estimans has come in, I think there's better ways of trying to estimate what would have happened if you followed the treatment as actually directed. I don't like breaking randomization. I like to take the strength of randomization in my statistical inference and my analysis. We've discussed an interesting one. Supposing, for example, a site has to close down, or one part of your study has to close down. Is it allowed? I'm interested. We have a panelist. Normally, I like all randomized, but if a site or an area of our trial closes down totally because of the situation, they're busy doing other things, can we just exclude them from our analysis? Is there a bias there?
I think it might. I think there could be a bias. If we excluded them, I can imagine a conversation going on that we're worried about this particular site. Seems to be a site issue, and we know that the site was in Madrid, who's been particularly badly hit by coronavirus. And by the way, there seem to be very few responders to treatment there anyway. It's all blinded. I don't know who's on which. Now, Paul, I can see you're laughing, but people do this. So I can imagine there could be some bias introduced by a little bit of sort of strategic choice of what constitutes a site that needs closing and which doesn't. I think you were thinking about the issues about the randomization. And I think if the study were stratified by site, taking one site out completely, I think would maintain the randomization, at least enough, given the other issues going on with this crisis. If it wasn't stratified by sector, then you've got a problem there. But,
your concern is how subjective, what else can influence the decision of removing people? And that it never gets documented. It's just happens in a conversation, and then the documentation is because this centre is clearly suffering a lot from COVID-19, and therefore we're going to remove them. So but then the whole per protocol discussion, if you're a fan of a per protocol analysis set, similar things can happen. Patients aren't included in per protocol. You find a way to exclude them. You don't document it, but you find a way to exclude them because you don't like what you see. I'm not saying. Yeah.
Yes, that may happen in your company, but not in mine. But at least then you're talking about an individual patient who hopefully the blind is still in place. And so this patient did particularly well or particularly badly, and you don't know if they're on active or control. Whereas if we're talking about a whole centre, and across the whole centre there's been very few responders, then we know that even in the active treatment group there are very few responders.
There's also few in there's few responders in the placebo, so the treatment effect itself may not be affected by removing it.
Well, it may or may not, but if you're looking for a given number of events to do your analysis, and even blinded, you haven't got many events coming from there. So it could have some impact.
I have other questions from the audience. If overall survival is our primary endpoint, should we be worried about deaths due to COVID-19? Would it not only be a problem if there is imbalance in death due to COVID-19?
So perhaps I'll pick this one up. If your endpoint is overall survival, and you have a target number of events to go to, and some of those events are COVID-19 deaths, then presumably any treatment effect may not have been possible in those COVID-19 deaths. So there may be, you may not be able to be picking up the differences that you wanted to pick up at the outset. But that, I think, is only the case if the COVID-19 deaths is a large enough proportion of the overall events.
Yeah, I don't know. We've got a feel for how big it is.
Go ahead, Paul.
I was just saying, I don't know if we've got a feel for how big an issue this is yet in our studies, how many COVID-19 deaths are occurring or are going to occur.
It probably depends a lot on the trial, the specific trial you're doing, the patient population that you're in, the age of the patient, the number of comorbidities. But there may be studies at that intersection of age and comorbidity where events could be non-negligible.
Okay, thanks.
I was going to add. There's an echo. But I was going to add that. Have to consider an analysis where a known COVID death is treated as a censoring time to the overall survival and compare that to the analysis by taking the overall survival as we know it.
Okay, we have another question from the audience. Sorry, we are trying to fix the issue of the echo, trying to find a source. What about quality of life assessment, healthcare resource utilization questionnaire, which are probably hugely impacted by the situation? Should we just not consider them during the COVID period?
I think one thing I've got sort of two comments to make on this. One is that we should be trying to collect data, but accept that we are going to have to look at those same endpoints in patients collected before this, during this, and after this, and see if there is consistency. And there may not be consistency, and that will be a problem for us. But the other point that I'd make is slightly different, and that is whether it's a quality of life endpoint or anything else for that matter, and I'm not picking out quality of life particularly, but we might need to start thinking about prioritizing what we actually do measure. And that may in some cases be a quality of life scale get taken out because we can measure other things that are more useful to us. And so I'm not sort of picking out quality of life particularly there. I think we do have to think about trying to trim down trials, trim down how much data we're collecting, and in some situations just go for really pretty good things. But that would depend a lot on what the indication is.
Thank you.
We haven't yet discussed, of course, the phase of the trial as well. You know, depending where you are, it can depend what you're going to change and how you're going to use the trial to make future decisions. If it's an early phase trial, perhaps we can change what we're collecting and minimize what we're collecting, not use regulatory approved endpoints and still gain information to move forwards.
Yeah. We have already a number of questions from the audience, which brings us to the next topic of regulatory interactions and impact on regulatory submission. The first question is, what are the requirements from IRB, EC, and regulators to change visit to virtual or telehealth? Have you seen any willingness to modify the typical lead time to review the submissions?
I can't address that directly, but I would say that all the regulatory agencies are under a lot of pressure these days for a lot of different reasons. One of the first priorities, of course, is to focus on investigational products for the treatment or prevention of COVID-19 and the diagnostic tests and antibody tests for that. Within the US, there's something called emergency use authorization, which could be used to fast track those kinds of things. It is clear that there are lots of clinical trials for COVID-19, and Cytel has a clinical trial tracker which tries to keep track of them. Having said all that, the FDA and EMA have come out with some guidance documents, I think in record time, having to do with what to do in the situation of COVID-19. On March 18th, FDA released their guidance, and Cytel followed up with a response the next day to that guidance. EMA on March 20th had a guidance, and Cytel had a response to that as well. On March 25th, the Committee on Human Medicinal Products Biostat Working Group had points to consider for methodological aspects. EMA followed that up on March 27th by version 2 of their guidance. And on April 2nd, FDA updated its guidance, including frequently asked questions. So that doesn't really address the question at hand, but it sort of gives you an idea of the fact that the regulatory agencies are acting at lightning speed, in my opinion, to try and figure out what to do with existing trials, what to do with new trials. What is clear is that in terms of regulatory involvement, that protocol modifications are going to happen all the time, and that the regulatory agencies need to be informed of that, and that will have impact on the statistical analysis plans and probably cause them to be updated accordingly. There are going to be lots of protocol deviations, I suspect, for all different kinds of reasons, and protocol amendments would be one way to address that, as well as the statistical analysis plan. But perhaps someone else could address the specific question that the audience member asked.
Actually, just a comment that I'd make to add to that. The regulators certainly have been moving at a very fast rate, seemingly come out with more guidance documents than there are patients. And every day there's more and more of these coming out. I'm pleased to see, but I'm a little bit worried. The FDA has talked very much about, talking to them about how you're going to solve these problems. And CHMP and European Medicines Agency has got an expedited approach to scientific advice, but still, these are kind of tractors, and we need racing cars. And I think the reality of being able to go talk to CHMP Scientific Advice Working Party, or go and talk to the FDA, the relevant division there, is going to be a real struggle, because there's lots and lots of people trying to do things, which is good. But I just wonder how these agencies are going to be able to react fast enough.
We have another FDA-related question. Considering the number of unconfirmed cases of COVID-19, should the FDA provide some special leniency for safety analysis during this period, especially that examines respiratory and fatal events?
I think the FDA guidance document tries to address that in terms of safety concerns and the notion that there could be other ways to gather safety information than was specified in the protocol. And James, I think, mentioned virtual visits, and so it's a matter of trying to be as flexible and pragmatic as possible, and that's what I think the regulatory agencies are striving for. And what that would mean is having some conversations, and I know, Simon, you're right, it's going to be difficult to have those conversations with regulatory authorities, but that would be the goal. I think it is a real good idea, the extent to which you can make it happen, to have conversations with FDA. In some cases, the modifications are going to happen before there is buy-in from the regulatory agencies, and the FDA guidance document allows for that provision in this time.
Is it feasible to assess? I mean, we have to produce safe drugs. We know that, and we have to assess the safety of our drugs. But is it feasible that primary safety assessment of these drugs will use data not during the COVID time or unaffected by COVID? So when we present all data, but primary safety doesn't use that because of the potential, or is that not feasible?
Question, Paul. I think that the Cytel response to the EMA document suggests that you could think about using historical information or some other source to get some idea of what to augment any safety information that you might have in the trial.
But I think also there's a possibility that more safety data, a very likely possibility, more safety data will be required after the product is authorized. And I mean, the classic situation in Europe would be conditional approval, where the reason for giving that conditional approval is that the benefits of putting a drug out there on the market, although you're still not necessarily completely comfortable with all of the data being adequate, the benefits of putting those data out there outweigh the downsides of sitting and waiting until we've got more information. So I think there's already regulatory mechanisms by which that can happen.
Another question regarding the pivotal trials. If we decide to amend the protocol to over-enroll so as not to include those impacted by COVID-19, could we change the intent to treat population for the primary efficacy analysis to exclude those subjects which are impacted by COVID-19, which would be dealt with the protocol population analysis under normal circumstances? Is it a legitimate approach?
I mean, as we've discussed a little bit, the difficulty about excluding patients is whether you're treating all treatment arms the same, or by excluding patients, you're introducing a bias either known or unknown. We can certainly increase the population size if we think the signals are going to be harder to find. And I think we could talk a little bit about interim analyses in a minute. Whether I would actually be comfortable about excluding a whole set of patients. I mean, we talked about the site issue. Can we exclude a site? And Simon already raised issues where maybe that's not a good idea. So excluding a whole bunch of patients as your main analysis could cause issues.
I don't think you would be able to present your primary analysis as this subset and just leave it at that. You're going to have to show the analysis of everybody, and or you're going to have to show the analysis of what happens in this subset that you decided to exclude. So everything is going to have to be put in there in some way or another. What you choose to call primary, somebody else may choose to call supportive. And then, as with pretty much every regulatory decision, it is ultimately the totality of evidence. Nobody gets an approval just because they got P with 0.049 on one endpoint, and therefore no other data needed to be looked at all. No safety, no secondary endpoints, no subgroups, all irrelevant because we got 0.049. That never happens. So it is always a totality of evidence type of discussion. And I think that's what would happen in this case if we wanted to look at that.
Okay, we have questions coming from the audience, which leading us to the next topic of role of the DMC. The first question we have, when to use or not a DMC to provide recommendation on the impact of statistical analysis,
So I'll start that. Talking about perhaps unplanned interim analysis. And you could do a blinded analysis at any point of time, but I think, because for other reasons, one might have to anyways consider protocol amendments. Perhaps there lies an opportunity for unblinded, unplanned interims. So for example, trials that probably would have gone, the path of having interim analysis, but they didn't because the recruitment was quite fast. This is because of this delay, there lies an opportunity to perhaps introduce unplanned interim analysis. Obviously, the statistical validity has to be maintained, perhaps using p-value adjustments or alpha spending functions. But anyways, it gives us an opportunity to look into the unblinded data, perhaps make some predictions, perhaps look at population enrichment, perhaps even look at what is the impact of COVID trials. For example, let's think about a trial investigating mental health. Now, mental health, we know, even for normal people sitting at homes, not going through our daily routine activities, that could impact. What kind of an impact? They can look at the interim data, gauge the impact of COVID under looking at different aspects. So that gives an opportunity. And I think, Bayesian methods here are particularly well suited, because then you can look at the totality of the evidence and with your prior belief and make some predictions to see, particularly if trials are nearing the close, is it time? Is it enough? Do we have enough evidence to stop early? Or if trials that have pre-planned interim analysis, and they are close to the timing of the interim analysis, is it already time to carry out that interim analysis? So these are some of the opportunities that could open up.
Yeah, let me add to that too. I can give you, well, I'll make it a hypothetical example in case there's anybody on the call from the company that I'm thinking about. I'm on a data monitoring committee, and this has genuinely happened, but I'll anonymize it somewhat. Let's assume that we were going to recruit 1,000 patients, and after 750 patients, there was an interim analysis planned, pre-planned, appropriate alpha stopping rules, and all the lovely stuff that we like. And this study has got to about 700 patients. So it's not quite there, but it's now stumbling because it can't recruit and it can't follow up patients and so on. So can we do the interim analysis early? Well, the sort of discussion that I had with some other statisticians around this was the data is not very clean. So we don't want to make a decision about stopping the study for positive efficacy reasons, hit the endpoint when the data is all very dirty. And I said, well, the DMC will have to consider that in their recommendation. And we then got into the sort of situation that I suggested earlier, where we might document decisions for one reason, but actually make them for a slightly different reason. So it might be that we've not quite hit the stopping rule. And so on the data monitoring committee, we say the data really weren't clean enough. So we think we need to wait until we do the interim analysis later on. And then we can introduce a bias there. But I think that we have to trust people who sit on data monitoring committees to make good judgments. I love to repeat a statement from the FDA's guidance on data monitoring committees. Actually, I hate repeating it because I wish I'd written it. But it said data monitoring committees should be encouraged to actually think about the problem and not just stick to the rules that are in the protocol. So it actually says, don't just stick to these things rigidly. And I think the DMC has to take that into account. And I would also actually quote something from the European Biostats group that put out their short page or so of guidance. Apart from an introductory paragraph, the first real guts of this, it says, most importantly, patient safety is paramount, the heart of every decision taken, regardless of any potential consequences of an ongoing trial. Type 1 error is extremely important, but it's not the most important thing. And if we have to kind of suffer a bit with saying, well, we're fiddling around with this, we don't know exactly what impact it has on type 1 error, that may just be too bad. We may have to live with that. is my moral ending to the session.
Simon, just to add to your example, perhaps stopping early may not be an option, but stopping enrollment might be.
Well, all sorts of things, or even making a call on futility, because keeping a study going in this current environment, if we're pretty sure that that futility analysis that was planned for 3 months' time is going to say stop for futility, actually we could do a huge amount of service by saying, yeah, cut it now, even though that was not when the futility analysis was planned. So it could go for positive efficacy, it could go for futility, but I think we have to have flexibility, and we have to trust data monitoring committees to make careful decisions, because they will be making some of these decisions based on unblinded data when all of us would sort of prefer that wasn't happening and it was done by a blinded steering committee or something like that. But these are special times, and we might have to do that.
We're actually coming towards the end of the timing of our webinar, and we have a lot of questions from the audience. So first of all, thanks a lot to our audience for being very active and sending out a lot of questions. I'm sorry that we could not address all, but we will follow up with you. We can save the questions and we will follow up. And you can also contact our COVID focus team and follow up with us with all your questions. So with that, I'd like to thank our experts for covering quite a broad spectrum of the topic and handling the questions we got from the audience.
Thank you very much. Goodbye.
Thanks, Natasha. Thanks, everyone.
Thank you. Thank you, Natasha.